Good afternoon, everybody. I'd like to welcome you to the third Retina Australia webinar for 2023, Vision Loss Priority Setting Partnership and an Introduction to Stem Cell and Gene Therapies. My name is Sally Turnbull and I am the Administrative Officer for Retina Australia, and I will be the facilitator of this webinar. As a participant, your camera and microphone will be switched off throughout. There will be an opportunity for you to ask questions once both our speakers have finished their presentations. If you have any queries during the proceedings, you can type a question to me in the Q&A section. Our first speaker this afternoon is Dr. Eden Robertson. Dr. Robertson is an experienced psychosocial researcher with a special interest in supporting children with a serious illness and their families. She has completed a Bachelor of Psychology Honours, Graduate Certificate in Adolescent and Young Adult Health and Wellbeing, and a PhD in Childhood Cancer in 2019. She is currently a postdoctoral research fellow at UNSW Sydney, focused for the past decade on addressing the psychosocial impact and challenges of several rare diseases, including childhood cancer, developmental and epileptic encephalopathy, and inherited retinal diseases. Alongside this role, she is also the National Research, Evaluation and Impact Manager for RedKite, a national childhood cancer support charity providing access to free, practical, emotional and financial support to help families take on the challenge of childhood cancer. In 2022, Dr. Robertson undertook a prestigious Fulbright Scholarship at St Jude Children's Research Hospital to further her skills in palliative care and best practice in supporting families at their child's end of life. She is passionate about empowering families to be more involved in their child's care, as well as involving consumers in research. In this webinar, Dr. Robertson will present on her Vision Loss Priority Setting Partnership and the process they will undertake to arrive at the top 10 research priorities for inherited retinal diseases. So I'll just hand over to Eden now.
Thank you so much, Sally. It's always a bit uncomfortable when someone provides such a lovely introduction to me. It's very humbling, and I'm very pleased to be able to present for you today. So let me just start sharing my screen. Okay, great. So to start with, thank you so much to Retina Australia for Julia Hall for inviting me to speak with you all today. I'm calling in from the lands of the Wallumattagal people, and I'd like to pay my respects to elders past and present. So as Sally had mentioned, I'm a behavioural scientist or a psychosocial researcher with UNSW Sydney, and over the past 10 years, I've very much been committed to how can we better understand families' experiences of rare diseases and develop appropriate interventions and support to ensure that they live the best possible lives as possible. So earlier this year, I was brought on to lead a project in inherited retinal diseases, and I feel very privileged to be a part of this role, as I'm able to partner with individuals with lived experience, health professionals and advocacy organisations like Retina Australia to be able to identify research priorities for inherited retinal diseases. So I wanted to take this opportunity to share a little bit about what this project involves, why it's so important, and then how you can support us if it is of interest to you. So as some context, and this may be all very basic knowledge for you, we know that inherited retinal diseases are the leading cause of blindness in children and adults of working age, and it affects around 1 in 3,000 people. And this includes diagnoses such as retinitis pigmentosa, Ushers, Stargardt disease, and Leber's. There have been some really rapid medical advancements over the past few years, and this includes improved diagnostic technologies and access to genetic testing. And at the moment, there are over 250 currently known genes that cause inherited retinal diseases. With the exception of a specific retinal dystrophy, the RPE65 associated retinal dystrophy, where gene therapy has proved successful, we know that IRDs are currently incurable. But it's really great to see that there's so much fantastic research out there at the moment, research into gene therapy and stem cell therapy, and it's showing promise for a future world without inherited retinal diseases. And you'll hear a little bit more about this from Vivienne after my presentation. So why is my project coming to existence? I think primarily it's because money unfortunately doesn't grow on trees. We know that we've only got a limited number of working days and days in a year to get this work done. And yet our list of what needs to be done feels quite infinite. So how do we actually decide on what gets shifted to that very top of our list? What is the most important area for us to focus on? What requires the urgency to get done now? And typically we've been driven by whatever the government, the funding bodies, pharmaceutical companies and researchers like myself decide what is most important and urgent. And how I like to describe this is using a bike path analogy. So imagine a square park, and we want to build a bike or walking path from one side, side A, to the other side of the park, side B, the opposite side. But you don't get any input from people that will be using that path. What you ultimately get is two very different looking paths. One is what the governments, the funding bodies, the pharmaceutical companies and researchers want. And then the other is what the patients, the carers, the family members and health professionals who care for those individuals want. And so these are really different paths, and it doesn't match up often a lot. So how can we scientifically capture what are the priorities for us as researchers? What should we actually be focusing on to ensure that the path that we're creating is the right path and the one that gets used? So the method or the approach that I'm using is called a priority setting partnership. And these projects identify and prioritize questions that have yet to be answered by research that key stakeholders consider the most important. And the reason being is we want to drive research that is going to be the most meaningful to those impacted by it. We want to ensure that we're walking along the path and creating the path that people want. So when we define our key stakeholders for this priority setting partnership, we have individuals who are living with an IRD, and that's including syndromic IRDs, such as Ushers. We've got guardians and family members such as parents, partners, grandparents, siblings, as well as the health and supportive care professionals such as ophthalmologists, GPs, educators, clinical geneticists as examples. And we want to capture the voice of all of these individuals to end up with these top 10 research priorities. And it's a bit of a mouthful of what we're including, but kind of broken down, we want to have a look at the priorities regarding anything to do with the prevention of inherited retinal diseases, the diagnosis of diseases, disease progression and control, treatment, as well as the management of IRDs. So it may be that the treatment-focused research may not be the priority for a lot of stakeholders, and that managing living well with an inherited retinal disease is more of an important focus. Or we could find that research into a better diagnosis pathway is considered more appropriate than research into disease progression. So this is all what we're trying to find out from this project. And so how we actually get to those top 10 priorities is this kind of roughly put a 5-step process. The first step is to create a steering group of individuals to guide how this project is completed. So rather than being directed by me, someone who has no lived experience of an IRD, I am guided by the people who understand these conditions and can advocate for the community on their behalf. So our steering group includes some fantastic individuals who have a child with an IRD, like Holly Feller and Emily Shepherd, who are the co-founders of Usher Kids Australia, and parents of a child who has Ushers. We also have individuals in our group who themselves are living with an IRD, such as the wonderful Leighton Boyd, as well as organisations who can meaningfully represent individuals impacted by IRDs, such as Retina Australia. And we've also got some researchers and health professionals involved in this project. I think I saw Lauren Ayton was online, and we're really grateful for their involvement to be able to drive through this project. We've also connected with some key organisations, and this is going to be really critical to allow us to not only promote this project and get participants or individuals to participate, but it's really to also disseminate the priorities to make sure that whatever we identify as the top 10 priorities is actually taken forward. And some examples of the key organisations that we're working with include Retina Australia as one of our key partners, Blind Citizens Australia, Deafblind Australia, and the Royal Australian and New Zealand College of Ophthalmologists. So we've got this fantastic group of people who are really committed to progressing the right research in inherited retinal diseases. The second step is now to actually undertake a survey with individuals impacted by an IRD, as well as parents and guardians and health professionals about what questions they have about the prevention, diagnosis, progression and control, treatment or potential treatments, epidemiology, as well as the management of inherited retinal diseases. So it seems quite complicated and quite broad, but really what we're asking people is to submit any questions they have about IRDs that they have unanswered, that they just haven't had that answer and they're still a bit unsure, and we'll really take it from there. So this survey is anonymous and expected to take about 5 to 10 minutes to complete. It can take a little bit longer if you're a bit unsure of what questions you want to submit. And there's multiple formats for people to participate, including over the online, and the QR code on screen can take you to the online survey. We can do it over the phone, on paper, on Braille, or in Auslan via a video call with the Deafblind Australia team. So basically, once we get all of these submissions, people are telling us what they have unanswered, what they're a bit concerned or wondering about. We collect all of these questions and organise them and work out which ones do actually still need research to answer. And what we're hoping to do is, once we get more responses, we will have a really representative idea from the Australian population about what questions are unanswered. So far, we've had 46 individuals respond, about 100 submitted questions. And we've got questions like, will I go completely blind? How can I slow down vision loss? Do lifestyle factors like diet affect the progression? And what clinical trials are currently available? So they can be quite broad questions, but this is the type of questions we're really wanting to find out what people with an IRD are wondering they don't feel like has been answered by research. The third step of our process is to take the most common questions that are submitted in that survey, the ones that are unanswered, and then ask individuals to rank these of these what they think is the most important. So imagine we've got a list of 40 questions, and now we want people to tell us what are the most important of those. What top 10 do they think are most important? And that survey is expected to go live around March next year. We'll take the top ranked questions then to a workshop, and these workshops will be a fantastic opportunity for individuals with an IRD, family members and health professionals to come together to discuss why they think certain priorities should be at the top and other ones perhaps not, to be able to finalise whatever we put forward as our top 10. So I think this is a really unique process to priority setting partnerships. And it's different to typical research, is that it's not up to me as the lead researcher to make that final call. It's up to the individuals who represent the community, who have the lived experience, who will decide what is in the top 10. And then we'll be opening up expressions of interest for those workshops around May, June next year. Then the final step is then to disseminate our top 10. So whatever our top 10 is what we will push forward. And we need to be taking that to funding bodies, to researchers, to organisations, to our partner organisations, to really advocate for this research to go ahead. And this is perhaps the most important part, as we want to make sure that those priorities are actually taken forward and actually undertaken by the research community. So we will be talking to funding bodies, we'll be trying to establish grants that address those priorities and advocate to researchers to undertake those projects. So to wrap up, the Vision Loss Priority Setting Partnership, it seems like lots of steps, but really it's just a way for individuals who represent inherited retinal diseases, who are impacted by inherited retinal diseases, to drive forward what actually gets done. So we will advocate for the top 10 research priorities, irrespective of what they are, whether they align with my beliefs, whether they align with our steering group's beliefs. We will advocate for those to the government, to funding bodies and to other researchers. And our hope is that the researchers will undertake these projects that address what is most important to those impacted by inherited retinal diseases, which will lead to more meaningful research. So if you do have any questions about this project, I do ask for you to reach out to me. You can email me at visionlosspsp@unsw.edu.au, or you can call me on 02 9348 0708, and we can talk through it. The survey one, where you can submit your questions, is currently open, and we would love to have as many of you participate as possible, and then potentially participate as well in the second follow-up ranking survey and the workshops. So I'll finish up there, and obviously putting some questions through to Sally if you have any questions now, and we can answer them after the next presentation. So thank you.
Thank you so much, Eden. That's fantastic.
No worries, thank you.
So we'll have some questions after we've heard Vivienne speak. So we'll do all the questions at the end of the session, if that's okay with people. And so now we'll move on to our second speaker, who is Vivienne Kaiser, a PhD candidate from the Children's Medical Research Institute in Sydney. Vivienne is a PhD student, and she's working under the supervision of Dr. Anay Gonzalez Cordero, who's one of our currently funded researchers. She completed a Bachelor of Science, Bachelor of Advanced Studies at the University of Sydney with first-class honours in 2021. She currently works on Ushers Syndrome, an inherited disease that causes deafness and blindness in children. Her work aims to develop AAV gene therapies for this disease using stem cell organoids derived directly from patients with Ushers Syndrome. This afternoon, she will provide us with an introduction to stem cells and gene therapies, touching also on her own project, focusing on Ushers Syndrome. So I'll hand over to you, Vivienne. Thank you so much.
Thank you, Sally. Let me just share my screen. All right. So hello, everybody. I'm Vivienne, as Sally mentioned, a PhD student. So I've just started. I'm in my first year in Anai Gonzalez-Cordero's lab at the Children's Medical Research Institute. So I'm just going to be telling you about what we do in the lab, pretty much. And so a stem cell story. We want to try and treat blindness with stem cells. And so just a brief background about our lab. We have 16 people, 5 PhD students, all under the supervision of Anai. And we are experts in stem cells. So Anai got her PhD from the University College London, where she was able to establish a stem cell facility when she came back to Australia, and it was the first of its kind in Australia. So it's really exciting research, which I'll tell you about. So first of all, what is a stem cell? So if you're unaware, stem cells are basically cells that have not been differentiated yet into an adult cell. So they don't know what they want to be when they grow up. They could be a gardener or a barista or personal trainer or a nurse, or they have no idea what they want to be. And so once they differentiate in what they want to be, that's where we get the adult cells from. So basically, you've got your stem cells and they can differentiate into whatever cell type. And so blood cells, muscle cells, cardiac cells, any single cell type. And it's quite amazing because it comes from the same cell. Everything in your body comes from the same cell. And so, our bodies are made up of 30 trillion cells, which is a huge amount, with over 200 different types of cells, and they all come from the exact same cell. And so, but where do they come from? Well, they do come from embryos. So after an egg is fertilized, in this stage here, which is called the blastocyst stage, these stem cells start to form and become and can go on to become whatever cell they want. Okay, so we can take them from embryos and we can grow them in the lab and differentiate them into whatever cell type we want. But it is obviously not very ideal to be taking stem cells from embryos. And so this amazing man in 2012 called Shinya Yamanaka invented a way to turn adult cells back into stem cells, which bypasses any embryo work at all. And so basically we get these adult cells and we put them back into stem cells where they can go on to differentiate into another type of cell. And this just requires 4 genes that can untrain our cells from being specialized. And so this means that we can take cells from a patient. So we can use blood cells or skin cells and turn those cells into stem cells. And so the advantages to this, again, is we don't need to use embryos for this. We can take cells from any patient, such as patients with diseases, children, and patients with different genetic makeups. And so what our lab is focusing on is using these stem cells derived from adult cells to cure genetic blindness. And so the most common causes of blindness in children, as I'm sure you're very aware of this, inherited retinal diseases in children, and then after 40 years, age-related macular degeneration. And so how can we, sorry, I can't read my screen anymore. So basically, we will give these cells instructions, these stem cells, to become an eye cell. Okay, because as I said before, these stem cells can become whatever you want. So when we give them instructions, they can become an eye cell. Okay, and we can culture these eye cells, and this is a really great advantage because it is like the eye is a very, very complicated organ, and it is, you can't just easily take eye cells from a patient or anything. So, but if we can differentiate them from stem cells, we can grow them in dishes and flasks and use them to study. But again, eyes are very, very complex, and just studying one different cell type in the eye won't actually give you an idea of the entire disease in as a whole. So what we want to do is we can group these stem cells together and add different things into them, and they can form mini organs or mini retinas. So they instruct each other, and they start to begin to form groups of eye cells, which then go on to instruct each other more. And we're left with a 3D mini human eye structure, okay? And so we've got in our lab, we make mini human retinas. And so, yeah, this is the back of the eye, which I'm sure you're aware that contains all the light sensing receptors in there. And so this is the key to vision. So we can form the cells necessary for vision in the mini human eyes, and these are functional retinas as well, with neural activity and all of that. And so, yeah, we can use these to study disease such as age-related macular degeneration or inherited retinal disease. And so one of the things our lab is doing, me specifically as well, is gene therapy. And basically what I do for this is we grow up the organs, the mini retinas, from patients with inherited retinal diseases. And so they have this mutation. We then can deliver a healthy gene to these cells or to these organs with a viral mechanism. Okay, and so for this virus, it's a safe virus because we have ripped out all the guts of the virus that is bad and replaced it with the healthy gene. And so this can go into the cells, and the retinal cells will be restored in the organoids. And so I'm doing this on Ushers Syndrome, where I'm creating some different gene therapies for that. So I've created 3 gene therapies for that, and I'm in the process of testing them out on retinas or on 3 different cell lines, so 3 different patients and their retinas. But I'm also using these stem cells to grow inner ear organoids as well. So I'm doing that in parallel. So another thing that our lab does is cell therapy. And basically with this, we get a healthy patient this time. We get the blood cells and the stem cells from that healthy patient. We grow up these organs, and then we isolate these photoreceptor cells from those mini organs and transplant them back into a patient with AMD or something like that. And then that restores vision. So we have done that already in mice. We've been able to get stem cells and remove the eye cells from it and inject them into blind mice, and vision has been restored in these mice. So it is a really promising approach. So yeah, so we're one step closer to curing blindness with these exciting mini organs. So now we can grow mini organs outside of the body and use them to create gene and cell therapies. And this is much better than using animal models or anything like that, because it is precise to that person and to that patient. And it recapitulates the human state very, very well. And so in the near future, hopefully we are going to use this stem cell technology to restore vision. So yeah, thank you for listening. And I would love to hear some questions or any comments, and just a thank you to everything that you guys do as well for our research. We really appreciate it. Yes.
Thank you so much, Vivienne. That's such an exciting presentation. We now have time for some questions. So if you'd like to ask a question, you can type it into the Q&A section at the bottom of your screen, or if you raise your hand, we can say your name and turn your microphone on so that you can ask your question directly, and then we'll turn your microphone off afterwards. So our first question is from Julia Hall. You should be good now, Julia.
Hi, can you hear me? Sorry, I can't turn the video on, but
That's it.
I guess we've got Vivienne here first. Vivienne, thank you for a very, very interesting presentation. I wanted to ask if somebody wanted to participate in a trial and have their adult cells converted to stem cells to participate in preclinical research, how would they go about seeking out to participate in the program?
Yeah, so that's a great question. You would probably contact my supervisor, so Ana Gonzalez Cordero. You can email her directly, but it's not like a straight process. There will have to be, there's a lot of ethical or ethic regulations and all of that that have to be undergone before your blood can be given. But it is definitely possible. We're always wanting more cell lines, because it allows our study to be a lot more holistic, and we can get an idea of different patients and the differences between them as well. So yeah, we do welcome people at all. So just email Ana Gonzalez Cordero.
Thank you, Julia. I had a question actually for Vivienne as well. You mentioned that you can treat the cells when you have them as stem cells. You do things to them to make them develop in a particular way. It's probably very complicated, but how do you make them become eye cells as opposed to a muscle cell? What is the process that you go through to specify that? Yeah, no, that's a great question, and it's still one that we're still because it's very new, this field, so we're still having a bit of difficulties with that. But basically, we mimic a baby growing in the womb pretty much, and there are certain genes that are expressed to allow these stem cells to differentiate different ways, and so we just give in a lab setting, give them those exact same factors and it's exact same concentrations. It's a very, very fine balance to get them to go to retinas. But basically for this one, what we do is, so we grow these cells, and then they grow like a monolayer of cells. And basically after a month, when we add all the different media that contains the factors specific for retinal differentiation, they just spontaneously just produce, and then we have to sit there and pick them out of the plate and transfer them to different wells. But yeah, it's really exciting that we can do it. And yeah, I've recently been able to create the inner ear now, which hasn't been done yet, so that's very exciting. Wow. So there's a very specific sort of recipe, I suppose, of what you need to put in. And how do you find out what that is? Is that just because you've studied the embryonic cells so much that you know what those factors are? Yeah, exactly. Yeah. And I mean, this research has been going on for a while. So I mean, I'm new here. The protocols were already established, lucky for me. But yeah, you start off by looking at how the embryo is formed, like in the womb, and that's already been very, very well characterized, from years and years ago. So we know the factors that are used, but we don't really know the balance of the different factors and the concentration. So it's just a lot of trial and error. And I say we get those retinas spontaneously pop up. We also get brains popping up, and we don't want the brains, but yeah, so it can be a bit. You just have to choose the ones that are doing what you want them to do. Exactly. Wow, isn't it? Fantastic. Okay, we have a question now from Leighton Boyd.
No, thanks, Eden and Vivienne. I just can't. I know Eden did a great presentation, and it's, as a member of that group, it's just amazing work that I think all of us, and I think as patients, we need to pass that survey on to as many people as we can. We need to know all those questions. There's no question that's a silly question. It's all important information. So I just want to really emphasise to people to pass that survey on, give us input. That's important stuff. And Vivienne, your work, I've been fortunate enough to go to the lab in Sydney, and it's mind-blowing, you know, the work that I and the team are doing. Can you just tell us a little bit? We keep saying we're growing, we're doing this and doing that. What are we growing them in? Are we growing them in the garden? Are we growing them in a saucer? How do we put them here? Store them into a special oven, or how does that work? Thank you.
Yeah. So we don't grow them in the garden. We grow them in our tissue culture lab, where we have to like properly scrub up. We have a mask, a hairnet, gloves, everything. It is going to be extremely, extremely sterile. We can't have any bacteria contaminating these organs, because they take 30 weeks to grow or to mature. And so if like bacterial contamination comes in at like 28 weeks, it's like, no. But anyway, so what we do, we keep them in incubators. So 37 degrees, which is the same as our bodies, because that's how they like to grow. So I think we have like 12 incubators and like 6 fume hoods where we do our culture. And yeah, it's great. A lot of work these cells take to grow, but yeah.
Very exciting. Thanks very much, Leighton. I have a question for Eden. Eden, I mean, obviously you've had some responses from people so far, and obviously still. Have you been surprised by any? Have you had any that you really weren't expecting?
It's been really interesting to see the questions come through. I think for me as a psychosocial researcher, I'm always kind of front of mind is how can we improve the well-being of families and you know, the mental health and adjustment to life with vision impairment. That seems to be a question that's coming through quite a lot. How can we actually manage mental health, especially when we know that our vision may be kind of vision loss can be progressing? That seems to be a question that's coming through a lot that I wasn't necessarily expecting. I think the way that the survey is set up is that you can submit 5 questions. And so it's quite broad, because inherited retinal diseases, even you can hear from what Vivienne's talked about, we kind of there's not enough information out there yet to really specify, I want to know this specific stem cell therapy and how it works for this specific disease. People are giving really broad questions, and that's really allowing them to provide 5 questions across 5 very broad topics. So I've got questions about cure, which we knew was going to be a question that's come up. How can we, you know, advance stem cell and gene therapy for a cure? But then, because that's one question in itself, and that's encompassing so many, for us as researchers, questions, it's allowed people to explore additional things like mental health, which probably is always set off to the side as a secondary research aim to cure. We've had some questions about how can we actually better support health professionals to support us as patients, to support the patients. Say, how can we better train health professionals to understand experiences of individuals with IRDs and how to better support them to navigate and to adjust well to life. So they're kind of the 3 main questions that seem to be coming out that are quite a clear theme. But as Leighton said, no question is a silly question. I think we're really wanting to get as many questions as possible so that it can be representative, so that whatever we get from the top 10 is truly representative of what the community actually wants.
Yeah, I was just going to say it's a wonderful opportunity for people to speak. Thank you so much. Julia Hall has another question.
Just a quick one for Eden. Eden, we have a lot of members who don't have access to online or digital facilities to answer the survey. I think you mentioned in your presentation that there's a phone number perhaps people could call. We get a lot of calls to the office where that's the easiest option. Is that your phone number on your presentation?
Yes, so that's my phone number, my personal phone number. And if you call it on either a Monday or a Tuesday, I will be able to answer it at that point in time. Or if you call outside of those hours, I will always give a call back, and you can complete the survey. We could just have a chat, and you can just tell me, what's kind of coming into your mind, and then I can kind of take your feedback and put it into the survey kind of structure, if that would be valuable. We can also do it via video call if that's easier, as well as having the Deaf Blind Australia consultant provide support through Auslan. And I can also provide it, mail out a hard copy paper version if that would be easier with screen readers.
That's terrific. Thanks so much.
Thanks, Julia.
Okay, so we've now got a question from Mark Boyd.
Vivienne talked a lot about ethics, and it's just a question on those ethical things. When you're dealing with creating life in different parts, what controls are there of how much you can create and virtually having a life and death sort of situation? Thank you.
Yeah, that is a very good question. Yeah, well, I mean, science is not at the stage yet where we can actually create life with these organs. They can't, like, these are, so for the retinas, it's just the retinas, it's not the entire eye or anything. But no, it is a really good ethical point that you made there. And I mean, I can't really answer it, I have to say. Yeah, it is tricky, but there is a lot of paperwork to get ethics approval to get someone's blood, and working with human samples. But yeah, no, it is a good point.
Absolutely.
Yeah.
Thank you.
Is there anybody else that has a question?
Sally, if it's all right, I might just comment in addition to what Vivienne just mentioned for Mark. That question that you just asked, Mark, about how can we ensure that, any samples provided and it's ethically used and not created life, that's the type of questions as well that my project, we're wanting to hear from people, because if we know that this is something that's really important for individuals, we will prioritize that and say we need to have protocols and information resources that patients actually do understand about how these processes work, and how their samples will be used and what it means for the future, and educating about what the ethics process actually requires. So Vivienne and I would have had to go through a really strict process where the project gets kind of written up in a really structured kind of document, then gets sent to external peer reviewers to look over it and say, well, actually, I'm concerned about the well-being of these patients. You need to provide more information here, or it needs to be explained more simply. This is the process that goes through research at the back end. That's often not talked about to consumers. And so if that's an area of research that we need to be ensuring that we're dedicating more time to, then that's the type of questions we're hoping to get within our priority setting partnership to actually dedicate funding to develop information resources to explain those things.
That's great. Thank you so much. I'd also like now to ask Rosalie O'Neill has a question.
I have the advantage of being able to be in Perth and Victoria, and so my brother passed away 4 weeks ago with Stargardt disease, and it took a lot of paperwork and a lot of ethical things so that his eyes have been donated to Associate Professor Fred Chen at the Lions Eye Institute. Is that an advantage to have tissue that does have the mutation?
Yes, of course. Yeah, it would definitely be an advantage. I'm trying to think of, like, if we can study exactly what he had and like the specific genetic mutations and all of that, and then understand more about this disease, then that is definitely beneficial for future people with Stargardt's. We are doing a lot of work on Stargardt's as well, but yeah, it is definitely helpful.
Okay, because they said that they'll keep the tissue for 10 years. And I thought, oh, that's pretty exciting, to know that it's going to be researched and looked at, and, maybe we can find a cure from that.
Yeah, for sure. So thank you for that. Yeah, it's very exciting to know that. But yeah.
Thank you.
Thanks, Rosalie. Leighton Boyd has another question.
Thank you. Look, I just want to tell the membership, ethics from Retina Australia's point of view, we're very, very, very conscious. We like to support research projects. We like to be the one-stop shop where, since Julia started, as Julia said earlier, we've reviewed our research protocol. Later on today and tomorrow, we'll be looking at some new funding applications. But at the prime, from our board point of view, we're always wanting projects and ethics approval, and I think it's important that that's the case. I think we do have a grants assessment committee. We have a lot of scientists and researchers on board, and I think that people and members should be rest assured that any project that we support as an organisation is well accredited, supported, and it's a very, very tedious and, as Vivienne said, it's a very painstaking process to get ethics approval. So I think that's great. I think from us as a patient point of view, we would be wanting to be, assured that the samples and the work that we're involved in as patients is all above board and ticks all the boxes. So that's just a comment.
Yeah, I think that's a really good point to make as well, Leighton, that any research you participate in, you have the right to revoke your consent from participation as well. So say if you consent blood to Vivienne's project and she uses that, if a year's time you go, actually, I don't feel comfortable with it, I don't want to include my sample anymore, if you contact those researchers, you can say, remove all of the data that you've obtained from my sample, I don't want to participate any further. And that's totally okay. There's no, there's not a black mark against your name for researchers or for hospitals. And so I think that's something that we really need to ensure that families do understand. If you do participate, that's completely up to you, and you can always change your mind at any point.
Thank you very much for that, Eden. Are there any more questions? I can't see any more, but you've got a final opportunity to ask a question before we thank our guests. No more? All right. Well, what I'd like to do is just thank both our speakers today for such interesting and exciting presentations. It's always so fantastic to hear about these new developments, and we can have so much optimism about the future. So I hope everybody has found the session informative. If you have any questions, if you think of any further questions, don't hesitate to contact us, and we'll make contact with Eden and Vivienne so that they can answer any queries that you might have. So that's the end of the webinar. Thank you both very much for your time and expertise this afternoon. And thank everybody else for joining us. And I wish you well for the rest of your day. Thank you very much.